Hello, hope you're having a great day. My name is On Wen, and in this video, I hope to share some findings about memory, learning, and the neuroscientific pillars underlying these concepts, particularly in light of the dynamic processes of information storage, consolidation, and retrieval. To this notion, I am covering the third to last section in chapter 6, the neuroscience approach, mind as brain, in the Friedenberg textbook. I wish to talk about memory because, nowadays our responsibilities and paths in life are getting increasingly complex and filled with, unexpected turns. And these wrinkles really hinder our ability to pay attention to the details for proper appreciation and internalization. And, by learning the science behind memory, I hope to attribute some credit to various parts of the brain that I'm not going to spoil, but are extremely pivotal in information retention. And through this notion, it would be great if we are cognizant of our physiological and psychological functions of those sections of the brain, and then, actively work toward maintaining a healthy brain in general for memory and other purposes, such as, solving puzzles like Sudokus or just vision, like our ability to see. So this lecture is divided into four principal parts. The first of which serves to bring everyone up to pace with some of the background information regarding how we sense our surrounding world and how memory is categorized into in the grand scheme of things. And then having equipped with those preliminary knowledge, we're actually ready to dive into the nitty-gritty central concepts of this section, which include the hippocampus, specifically its intimate relationship with learning and memory. In addition, the second half of today's video will allow us to have a closer look onto the neural substrates, that is, the brain areas that are responsible for generating and maintaining the two types of memories, and definitely related experiments and their interpretations. So first, let's get started on, bringing everyone on the same page, starting with sensory modality. So the first crucial background concept is this sensory modality, and it is an aspect of a stimulus that is perceived. This comes in five flavors: iconic or visual, echoic or auditory, olfactory or smell, gustatory or taste, and lastly, tactile or touch. Each of these forms can correspond to multiple representations because we as humans are exposed to a plethora of sensations and, as a result, evolve different levels of dealing with each of them. For instance, for iconic modality, you can perceive an object's sharpness, color, and even subtle shading. Let me give you another example in the case of echoic modality, in which this can manifest as the frequency, loudness, and pitch of a sound. And then we have memory itself. And memory is defined as the capacity to retain information over time, whether in the short or the long term. And we gotta perceive a stimulus first via the five sensory modalities that we just talked about and encode it. Then we're able to store and retrieve it as memory for later usage. Memories are actually grouped into um, two types according to what I call residence time in our head. The first one is short-term memories, or they're also called working memories. And the second type is long-term memories. And they, as the name suggests, reserved for an extended retention of information. So let's briefly touch base on these two types of memories, starting with working memory. And in this regime, information storage is relatively brief, averaging around 10 to 20 minutes. And even though it's only 10 to 20 minutes, it's actually still much longer than the memory that's related to our sense, which is only like a few seconds at most. But then on the flip side, it retains less information, just a small number of items. And there are actually three psychological components of working memory, and they are articulatory loop, which refers to the activity in which operations on verbal representations are performed. Visual spatial sketchpad is the visual aspect of it. And lastly, the executive control system, which helps to coordinate the activities according to the information received. But, as we know, we can store data in our noggins for much longer than that. And this type of memory is called long-term memory. And there are actually two types. And the first being procedural memory, which showcases in a person's skills through demonstrations. And the second being declarative memory, which relates to facts and events and can be assessed by, speaking or conscious recall. And something that I should mention here is long-term memory is usually stored for months or even years, which is super interesting. So having gotten on the same page regarding those four preliminary concepts, I think we're ready to explore learning, memory, and the hippocampus in general. So let's start with learning. So learning takes place when a particular event induces a change in someone's nervous system, which then provokes a change in their behavior. In other words, you can view this as a quote unquote nervous learning as a formation of a neural circuit, similar to an electrical circuit. So in particular, when the circuit is activated at some point, the memory associated with it is experienced and then informs that organism's behavior. So I like to think of this idea with an analogy with, an electrical circuit in which a light bulb, will shine if the battery sources are connected properly. Given that the circuit is, already there and established, the light bulb cannot just turn on by itself through, like sorcery or witchcraft or something like that. But yeah, Donald Hebb is really a pioneer in the 40s and 50s in this subcategory of neuronal changes called synaptic plasticity. So let me define this first. So plasticity um, is the capacity for structural or biochemical synaptic change. And there's actually a rule named after him that describes the connection between two neurons. In particular, if you fire two neurons that connect with each other, then their synapse will become strengthened. And so let's take a closer look at this long-term potentiation phenomenon. So let me define it first. So it is a specific change that mediates this, Hebbian form of learning. And it is actually the enduring facilitation of synaptic transmission that occurs after the activation of the synapse by intense high-frequency stimulation. So the key here lies at the rapid and the repeated parts of neuronal synaptic stimulation. So long-term potentiation has shown to not only increase the responsiveness of synapses upon activation, but it also increases the number of receptors in different regions. And the latter effect is shown here in this figure taken from an experiment in 92 by Talco. And specifically, they actually measured the binding properties of AMPA in form of optical density. So the x-axis here represents the different regions of the brain, and the y-axis, on the other hand, shows the optical densities with a higher value corresponding to more AMPA receptors in this case. So you can see for each triplet, applying a low frequency theta burst, which is the middle column here, theta, enhances the y values compared to the low frequency on the left side. So the first column, in a statistically significant manner. So you have the two stars and the one star here. But then after examining this figure, I actually would have loved to, see more on the explanation of this theta star wrinkle here. So like the third column, in which there's actually no significant increase compared to the first column, despite being like high frequency as the author defined it. So let me backtrack real quick. So the second column is theta. So theta here is theta in low frequency, but then the third column represents theta burst stimulation with a higher frequency. So there has to be a reason behind this, but um, when someone thinks about it, if you have more, if you increase the frequency of some type of stimulation, you would probably see a higher response, a higher response in the third column here, but apparently it is not the case. And it would have been nice to see some type of explanation in um, in a paper by the authors. But regardless, this research is paramount in suggesting the practical effect of LTP, long-term potentiation, in increasing the reception of neurotransmitter for sure. So now I think we're ready to talk about the hippocampus because interestingly, LTP has most often been studied in the hippocampal cells. And in 1984, there was this study, revolutionary study for sure, conducted by Suzanne Corkin with a patient called HM. And this guy, HM, he suffered from epilepsy, which is a disorder in which the neurons fire uncontrollably, which leads to muscle spasms and seizures. So then, because of the seizures, the surgeons would make sense for the surgeons to to end the seizures, right? So they actually decided to remove parts of HM's medial temporal lobes, including the hippocampus here, to stop the seizures. But I should note in this figure here that the surgeons actually remove both sides of the hippocampus, even though this figure only shows one. It's only for illustrative purposes. But yeah, while the removal actually was able to cease their seizures, HM actually could not learn new information, but his long-term memory prior to surgery was still there. For example, he could talk to his friend, turn around and talk to someone else, turn back and forget who the first person was, which was, kind of wild. And something else that is a bit wild as well is that he could actually still solve puzzles, albeit the same puzzles multiple times because he doesn't know that he's solving the same puzzle like all over. But this actually suggests that his problem solving capacities were still intact. And through these results and observations, the Korkin and her colleagues were able to interpret that his removed part of the brain was responsible for consolidating working memory into long-term memory. And furthermore, severing a particular brain structure can also remove a component of memory ability. And looking back, even though the strength of the experiment is that it directly conveys the main function of the removed structures, the researchers would be a bit hesitant to actually replicate this without distorting the ethics. So the replication side of the scientific method is a bit, at um, is being contested here, unless someone is medically indicated to remove something due to ailments by doctors, then they are able to conduct such experiments like this. But I think it's a bit rare, per se. So yeah, and so let's take a closer look into the hippocampus here. So the hippocampal system works in three-step process in which information about current events arrive from the cortex at the hippocampus for processing. Then the hippocampus sends it back out to the cortex for storage in a more permanent sense. So through this notion, one can think of the hippocampus as an integrator, which combines and unites different pieces of information into a single whole. And this is in direct contrast with the cortex, because the cortex operates through the divide and conquer fashion. So let me give you an example that revisits the concepts that we have discussed thus far. Suppose this girl right here is studying the slides for a test the day of at 2 a.m. Because the clock here says 2 a.m., which is a bit unfortunate. But you can see firstly, her visual system will register the image of the slides I put over here, while her auditory system will detect the sounds of maybe the video recording of the lecture, or like suppose there's like an owl outside of her window. Maybe she would be able to smell the fragrance coming from the plant nearby, alluding to the olfactory modality of the five modalities that we've been talking about. And in this case, the various parts of the cortex have divided this rather unfortunate studying experience into fragments. And then these fragments will converge as inputs to the hippocampus. And then the hippocampus combines them to form the whole memory of exam studying in sparse distributed code that retains its connection sensory cortices as well, which is pretty nice. So now let's take a deep dive into identifying the suspects that underlie the working memories and later on long-term memories. So story time. In '96, Ah and their colleagues performed an experiment in which the control population either searched for a single target letter or responded manually to the letter that was consistently rehearsed. So these controls serve to probe the storage and rehearsal aspects of verbal memory or memory in general, respectively. On the other hand, the experimental group viewed the letters and signaled if the letter they saw 2 cards ago matched the current one. So I just wanted to take this time to stress that, this latter activity here is a bit more sophisticated than the control ones in that it required both storage and rehearsal components. So in this figure over here, this first panel called two back, it corresponds to the experimental group in which the the participant will let the researchers know, um, as in a yes response, if the letter that they saw 2 cards ago, which is P, is the same as the current card, which is P. And yeah, for the search control, then um, there would be no response because the letter is F and 2 letters ago it was M. But the very next one, it was a yes response because they they view the letters and then they search for a single target. But then for this rehearsal control, the participants viewed the letters and responded manually to the letter consistently rehearsed. So in this case, you see the letter M over here, the very first card. You see the letter M, and then you keep repeating M, M, M until you see the very next word, which is P, and then it keeps going on and on. So yeah, after doing that, they did some PET scans. Not the dogs and cats for sure, but the positron emission tomographic scans to determine the brain activity. And at the end of the day, the purpose of this study is to identify the parts of the brain that are responsible to storage and rehearsal. So as a result, they noticed that the posterior parietal cortex on the left hemisphere specifically was largely responsible for the storage half of the equation, and 3 different areas for the rehearsal side of the equation, which are Broca's area, the region in the premotor cortex, and another region in supplementary motor area. And as you can see, they titled their title here as significant activation foci for memory minus control in the item recognition task. So I just wanted to um to bring you guys to this minus part because um this is important because the experimental group contains both storage and rehearsal. So to find true storage, you got to subtract the control storage, which was group one, storage group one, from the experimental population. Same thing for rehearsal. So something to keep in mind for sure. And yeah, and for spatial memory, and in '96, Smith conducted a somewhat similar study as all, but this time to identify some suspects that mediate spatial memory. And in particular, participants were shown letters at different positions around an imaginary circle. So to be clear, the center of this circle is the plus sign that you guys can see in the middle of the card. And then the participants had to determine whether the letter they saw 3 cards ago was the same as the current one, which is over here. So, and then, so So let me backtrack. The participants had to determine whether the card they saw 3 cards ago was the same as the current one or was in a different position. So, so for example, for this letter G, 3 cards ago, the letter was also G, so the response would be yes. Um, and on the top panel here, the letter they see currently is D. In the top left position of the card, and the participant should respond yes if the card if 3 cards ago the word was also in the top left corner, which was the case over here. Um, Even though the letter is G, that doesn't matter in that case. It actually turns out that the right posterior parietal cortex warranted the high Z-score of 7.4 over here, which was shown on the rightmost column in the figure here, and the 7.4 is here, and the posterior parietal on the right hemisphere. And through this notion, the authors were then able to conclude that this cortical region is responsible for short-term memory processing of space, so spatial memory. And so that was short-term memory and friends. And for this last section, we're going to get to know long-term memory and friends. And in 97, Varga and Kadim and her colleagues studied a group of patients um, who sustained only hippocampal damage early in life to find the neural substrates of declarative memory, which was arguably of theoretical importance. So in this case, the authors were trying to learn about the declarative memory. And as you can recall, the other type of long-term memory besides declarative memory is procedural memory. So something to keep in mind. But yeah, the names of the patients are shown over here, and they're Beth, John, and Kate. They actually didn't recall anything that happened early in the day. But then they performed pretty well in schools, as shown by these test scores. Um, and the moral of the story here is that the hippocampus proper actually mediates episodic memory because that was the part that was damaged. But on the other hand, the part called the limbic cortex mediates the semantic um, memory because it was not removed. And then it was able to mediate semantic memory in these individuals in that their test scores are pretty good. You have the most of the case, you have the predicted score um, over here, and the actual score is around that number to that degree. And going on to procedural memory, in 96, Nudo was hoping to search for the possible suspects that mediate procedural memory. His experiment was interestingly different from the previous ones we've discussed so far because this time monkeys are involved, so not humans. And in this case, monkeys were trained to, pick up small objects with finger and hand movement, so wrist, forearm, or another set of activities, finger extension, finger flexion, and wrist abduction. And these actions were actually found to enhance the activity of this middle area. So let me orient ourselves here. So the left panel um, is similar to like a heat map that depicts the brain activity of a representative monkey before training and after training for the right panel. And as you go toward the top of the slide, so going up, you're actually heading rostrally or to the front of the actual monkey. So the front of the monkey is here, and the back of the monkey is over here, similar on the right panel. And on a similar vein, as you go towards the right, so toward the right, you're heading medially or toward the middle of the monkey. Um, and same thing over here. And you can actually see that the color change happens most strikingly at this middle area here. So it was mostly, let me give you an example. So it's mostly blue in this little like left area at 7 or 8 o'clock. and kind of blue over here in the 4 to 5 o'clock position here, and they all turn black in this in the right panel here. And same thing in the middle, like there's like a yellow blotch here, but there's only like one smaller yellow area over here. So um so yeah, so the moral of the story is that this the area that exhibits the most amount of change is actually corresponded to the primary motor cortex. And so yeah, so the primary motor cortex is the main suspect for procedural memory mediation. And other researchers were also identified the basal ganglia as one of the main players in the learning of skills. And so yeah, something that I would do following experiment like this is to follow up and extend it to identify, further possible brain areas that play a role in these types of, procedural or declarative memories, or even working memories as well. Specifically, I would possibly do so by probing, other brain areas with other types of, activities to see if there's any variation in the results, which is kind of recorded here. Or let me go back to this figure by Smith here that shows different z-scores to see if there's any variations, and to see if there is any particular brain area that was not mentioned or was mentioned, but have strikingly contrasting numbers. Let's put it that way. So yeah, thank you so much for watching this video lecture. I really hope I was able to, shed some light onto the intricacies of memory, learning, storage, or even memory retrieval. And there's this meme that I put over here that's arguably relevant to our topic at hand here. Let me I'll just email this to myself as a reminder. Oh, look, a new email. I hope after this lecture, you'd be able to recognize this, that this refers to working memory or short-term memory. And this type of, I hope you're also, you can recognize short-term memory in that it is mediated by the left posterior hemisphere, the Broca's area, part of the premotor cortex, and part of the supplementary motor area. Um, so yeah, thank you so much. Thank you again, and I'll see you soon.